Enzyme regulation and metabolic pathways. My name is Alemi Michael. Welcome back. In our last lecture, we measured the speed of enzymes. Today, we learn how the cell controls that speed. Because life is not just chemistry, it is a controlled chemistry. What is an enzyme regulation, first of all? Inside every cell, thousands of reactions occur simultaneously. If left uncontrolled, chaos would result. So the cell uses regulation and mechanisms that turn enzymes on and off. Inside every cell, this regulation ensures efficiency, energy conversion, and a balanced homeostasis. Let us talk about metabolic pathways. A metabolic pathway is a sequence of enzyme-controlled reactions where the product of one step becomes the substrate for the next step. Types. There are only two types, namely anabolic pathways. These pathways build complex molecules from small ones. They require energy. And the second one is catabolic pathways. They break down molecules into smaller ones. Therefore, catabolic pathways release energy. These two pathways are controversial. They are the reverse of one another. Think of these as an assembly line where each enzyme performs precise steps. Number one, control mechanism. Feedback inhibition. One of the most powerful control mechanisms is feedback inhibition. The mechanism ensures that the final product binds to the first enzyme and stops the pathway. This prevents wastage. The cell produces only what it needs, nothing more than that. Control regulation number two, allosteric regulation. Some enzymes have special sites called allosteric sites, where molecules bind and change the shape of the enzyme. This happens upon need to control the end product. Now consider these two factors. One is activators. Activators are the substances that stabilize the active form of an enzyme, and they increase activity. Whereas inhibitors, they destabilize the enzyme. Therefore, they make the inactive form of one enzyme to become stable, and therefore they decrease activity of that enzyme. Allosteric enzymes often show cooperative behavior. Binding of one substrate makes it easier for others also to bind. Regulation method number three. Regulation mechanism number three is covalent modification. Enzymes can also be regulated by chemical modification. Phosphorylation, for example, where addition of phosphate group from ATP is formed, and this can activate other active sites. Phosphorylation, for example, where addition of a phosphate group is identical from ATP. This can activate or deactivate the enzyme. Next is a role of cofactors and coenzymes. Some enzymes also cannot function alone. They require helpers. So the helpers are cofactors, which are inorganic ions like zinc and magnesium ions, and also coenzymes, which are majorly inorganic molecules, often vitamins. For example, FAD, the flavinoadenine dinucleotide, NAD, nicotinamide adenine dinucleotide. Master these two terms. Apoenzymes are inactive forms of an enzyme, whereas haloenzymes are the active enzymes plus their cofactors. Now let us integrate metabolic control. No pathway works in isolation. They are interconnected, forming a dynamic network. The big picture here is that cells adjust enzyme activity based on the energy level and the requirement of energy, majorly ATP and ADP. Also through substrate availability and environmental conditions. A living cell, enzyme regulation is the language of life is control. It ensures balance, precision, and survival. Without regulation, life would collapse. In summary, enzyme regulation controls metabolic flow. The pathways are stepwise enzyme sequences. End product inhibition prevents overproduction. Allosteric regulation changes enzyme shape, while covalent modification acts as molecular switches.